Okay, so we're getting kind of towards the end of the module now. We've covered a lot of stuff about the genome and about medical conditions and how we can use genetic technologies to fix them. A lot of the rest of the stuff that we're going to be talking about is about genetic engineering specifically, and most of this is going to be on plants for the agriculture industry. But if you're interested in kind of the human medical side, it's still important because you're going to be covering a lot of principles that you need for like general genetic engineering. This is going to be a shorter one where we're going to be talking about Agrobacterium, what it is, what disease it causes in plants, and how it causes that disease, to give us an overview on Agrobacterium and its plasmids. And then in the next few, we're going to be talking about the plasmid specifically and why it's important and how we kind of use it in the broader scheme of things. So let's just get into it. Agrobacterium is a type of bacteria responsible for causing crown gall disease, which results in large disorganized tumor-like growths on plants. So the images on the right are just plants, and these look kind of clumps of cells that just don't look right. If you know how plants grow, obviously, they shouldn't look like that. They shouldn't have a bunch of kind of disorganized clumps of cells just in the middle of a branch or in that stem. So this is clearly not natural for the plants, and it's been caused by some reason. Agrobacteria, isolated from these galls, I'll just call it galls because it's easier, can induce new tumours when inoculated into the wound of a new plant. So if we take the agrobacteria that are causing this crown gall disease, so these agrobacteria have caused the growth of these tumor-like balls on the plants. If we take the bacteria out of these galls and we put it into the wound of a new plant, it's going to create a new gall, and it's going to grow and grow and grow, and then we'll have another one of these tumor-like balls on the new plant. The agrobacteria can even be removed from these galls, but the plant is going to still display the characteristics of a tumor, like rapid uncontrolled growth. So even if we take all the agrobacteria out of these galls, these tumor balls, they're still going to grow and proliferate. And that indicates that the agrobacteria have permanently transformed the cells in these galls, which we would imagine would mean that the bacteria have altered their genomes. So the tumor-inducing principle, which basically is trying to figure out why these tumors are being formed. So I'm just going to use the word tumor to describe the gall because it's easier, but just know I'm using the terms interchangeably. The tumor-inducing principle for crown gall disease was initially thought to be a toxin or bacteriophage that transformed the plant cells, but this turned out not to be the case. There are two different kinds of tumor produced by two different kinds of agrobacterium. The first kind of tumor produces these tumor-like growths from the plants, and within these tumors the plants produce a carbon source called octopine, so it's kind of like a sugar. And then the second type of agrobacterium, it also produces these tumor balls, the galls on the plants, but it produces a different sugar, nopalene. The octopine and nopline produced by the different strains are the carbon compounds the strains use as an energy source. So it basically just means that these different kinds of agrobacterium, they're infecting the plants, they're causing growth of the tumours, and within these tumours, the plants are being forced to produce these octopine and nopline compounds that the agrobacterium in the galls are using as an energy source, and then the agrobacterium are going to tell the plants to make more of the energy source so they can grow and proliferate and live. A scientist shown on the right called Jeff Snell discovered that a plasmid that we now call the TI plasmid was responsible for the initiation of the disease. Bacteria cause formation of the galls by the plant and production of the carbon compounds that the bacteria use as an energy source, like we just talked about. Snell found that the cured non-oncogenic plants he found that cured non-oncogenic plants, so plants without the tumours, could be converted into oncogenic ones, so they would grow tumours, via bacterial conjugation. Snell proposed the genetic colonization concept, where the Agrobacterium were producing TI plasmids, which are these you have the nucleoid in bacteria, which is their central genome, and then you have these very small circular pieces of DNA called plasmids. These TI plasmids are only 200 kilobases in length, containing very few genes, but they can be important giving bacteria resistance to stuff like antibiotics or to produce specific things. So the agrobacteria are inserting these TI plasmids into the host plant cells when they can get into them through the plant being damaged. These TI plasmids evolved for the benefit of bacteria, because when they inoculate them into the plants start producing the carbon compounds that the bacteria need to eat and live and grow. The crown galls are the result of the healthy plant being transformed by these TI plasmids. When introduced to the plant cells, the TI plasmids induce the host plant cells to produce these carbon-rich molecules that the agrobacterium are using for energy, called opines. The opines we talked about earlier, octopine or nopline, depending on what strain of agrobacterium we're talking about. And then the free-living agrobacterium within the galls are going to use these opines as an energy and carbon source to live and proliferate and then continue the cycle. So that's an explanation of agrobacterium and kind of how they're causing these tumor-like growths in the plants that we call galls, how they're doing that, and a look at the TI plasmids. So I produced these two images on the right here. The top one is the TI plasmid for the strain of Agrobacterium that sustains itself on the octopine, and then the bottom one is the TI plasmid produced by the strain of Agrobacterium that sustains itself on nopline. So it's important to remember the two different strains of Agrobacterium are producing two different kinds of TI plasmid to produce galls in their host plants. They're producing the two different kinds of energy-rich, carbon-rich molecule that they're feeding off, called the opines, octopine or nopline. So we'll go into how we use these TI plasmids in genetic engineering in the next one.